Good afternoon, everyone. My name is Mindy Christensen, and I am the Medical Director and Director of Fertility Preservation at the Johns Hopkins Fertility Center. I'm very excited today to welcome you to our special Oncofertility webinar. We are very pleased to host Dr. Sherman Silber. He's a worldwide pioneer in reproductive medicine, surgery, and fertility preservation. He will give us an interesting presentation today on making competent oocytes from skin cells. The webinar itself will take about 45 minutes. The presentation will follow by 15 minutes of questions and answers. Please keep your microphones muted and your camera turned off throughout the webinar to avoid side noise, and so we have a smooth webinar broadcast. If you have a question, you can post it in the chat. We'll open the discussion at the end of the webinar, and you'll at that time have the ability to turn on your mic and camera and ask questions. We'll record this webinar and it will be uploaded later today to our YouTube channel. So thank you very much for joining, and we are very excited to hear Dr. Silber today.
Well, thank you very much. Thank you very much, Mindy. I appreciate the introduction. We have a feedback, or is it okay? Yeah, I think it's okay. So first I wanted to mention just for a second that Johns Hopkins is very instrumental in what we're doing with ovary allotransplantation. You have some amazing people there. Dan Brennan was originally from St. Louis, and he's guiding us through this. And I'll talk at another time about allotransplantation from a sister or cousin to a Turner syndrome patient, for example, with immunosuppression that's quite safe. And we couldn't do that if it weren't for Dan Brennan and the Johns Hopkins team. So we appreciate that. So today I'm just going to talk about in vitro oogenesis, which I believe in 10 years for sure will be one of the routine things we'll be doing in IVF and in fertility preservation. And I'll explain to you why. And in humans, we're already halfway there. But it might be another 5 years before we figure out all the way, and then another 5 years after that, I'm guessing, for all the regulatory reviews that will have to take place. But I was just at a meeting last week where people were saying it's inevitable that in vitro gametogenesis is going to be eventually where we're heading to, particularly for so many women that miss the chance to have their ovary frozen or their eggs frozen before sterilizing chemotherapy, or before they just reach the age where they finally want to have children. So, oh, wait, I think I thought it worked before when we tried it. Let me try this. Yeah, okay. I started out working about 8 or 10 years ago with Katsuhiko Hayashi in Japan. And he was the one who first figured out how to actually come up with fertile oocytes from the skin, from the tail of mice that, female mice that had had their ovaries removed, and came up with healthy mouse pups. And now he's got many, many thousands of these healthy mouse pups derived just from the skin cells from the tail, the fibroblasts of the tail of these mice. So we're just translating his work. And this is Ori Hakabi, who no longer is with Katsuhiko, but she was originally the technician that did most of the work. And I was with them for so much of this, and they would work till midnight every night and then come to the lab 6 o'clock in the morning the next day, because it was a lot of work to originally do this pioneering study. And now we're working with Amanda Clark and Hayashi, and Amanda at the UCLA is working on converting these skin biopsies from our female patients that are postmenopausal and our male patients that are azoospermic, converting them to stem cells into PGCs and eventually to oocytes. And Kyle Orr will be the one that's going to be converting these PGCs into sperm. So this is our CHOSE team, we call it, and that's Kyle. You all recognize him, and you may not be familiar with Sierra, who's my scientific assistant. And there's Amanda Clark, and there's Hayashi, and there's our benefactor for this study. So you understand that we're going to be making fantastic oocytes because we're simply replaying early embryonic formation of oocytes. That arrow just points to the green stain of the initial cells in the epiblast that are designated and to become germ cells. They're not even germ cells yet, but they are designated to become germ cells. And we are going to just follow the normal transcripts that in the early embryo allow these designated cells to actually become PGCs, and then the PGCs to become oocytes. So early epiblast cells are separated from the rest of the developing embryo, and that makes sense because the rest of the embryo is going to be differentiating, and these cells can't be differentiating. So that's why they're literally outside the body. So in vitro gametogenesis just copies this early embryonic process. Now, think about this: oocytes generated in vitro from these iPS cells. We shouldn't call them stem cells, or they're induced pluripotential cells, but they are really stem cells. They'll be very young because we're just recreating embryology. So in the future, for example, a 38-year-old woman might be better off having a skin biopsy than an oocyte retrieval for IVF because we'll get younger eggs. And this is just what people probably already know and they're already familiar with. Let me go back again to that. Yeah, that the primordial germ cells are really by the yolk sac outside of the developing differentiating body. And then by 3 weeks of fetal life in the human, they start migrating up towards the dorsal ridge where there's either a testis or an ovary. And during this process, basically what's happening is totipotential cells are differentiating into primordial germ cells. And actually, interestingly, clinically, the differentiating cells, these early, you might call them stem cells or undifferentiated cells that are destined to become germ cells, these cells, the ones that don't differentiate completely into primordial germ cells and enter the ovary too soon, become embryonic rests that turn into tumors that are what we think of as hemartomas. And in the testes, these it's been shown in the Page lab that these cells that haven't differentiated fully into PGCs and enter as stem cells into the testes eventually are going to become embryonal or teratocarcinomas. Now, so germ cell development in mice is very important to understand so we understand the human. And this is a complicated diagram that you may want to study in more detail, but you can see that below is the blastocyst epiblast cells go to PGCs, they migrate, and in the male, they go through mitosis and they go through spermatogenesis, and they don't really go through meiosis till puberty. And for oogenesis, if they go find a female germ cell at 3 weeks when they reach the the ovary, they obviously go into enter meiosis right away because of stimulation from retinoic acid and upregulation of STRA8, but you don't have to know that at this moment. But they enter meiosis and they would all be gone by the time the female fetus is born, except that they become primordial follicles in response to the tissue pressure of the fetal ovarian cortex. And as primordial follicles, they become locked, and you only lose 1,000 a month based on tissue pressure release. And if it weren't for this locking of right after meiosis, the eggs would all be gone by the time the female is born. So you might wonder why nature created such a crazy system where in the testes you have spermatogonial stem cells developed from these PGCs that enter the fetal testes, and the male can make sperm forever, and why in the female they had to go through meiosis first and be locked in meiosis so they're not lost. Why would be the evolutionary advantage to such a crazy concoction? And Hayashi postulated that the evolutionary advantage is that without spermatogenesis, we wouldn't have mutations and we wouldn't have evolution. Evolution required spermatogenesis with lots of mitosis and the possibility for mutations. Whereas with oogenesis, you really don't get continued reproduction of these germ cells. They just stay what they are from the moment they enter the ovary in meiosis and get locked in meiosis. So you're not really going to get mutations. So the spermatogenesis allows for evolution, but oogenesis stabilizes the species. That's a philosophical way of looking at it, but it probably is really the reason why such a crazy system evolved. And so when we reconstitute PGC specification using mouse pluripotent stem cells, it looks somewhat like this. We're going to use the transcripts. And Hayashi figured out these transcripts originally, same way Yamanaka did, figuring out the transcripts for making iPS cells. You just do reverse transcriptase and you find out what are the active genes that are really working at the different embryonic stages. And the epiblast has very specific transcripts that turn these ES cells into epiblast cells. And then you have very specific transcripts that turn the epiblast cells into PGCs. And this is just a diagram showing that once you have the PGCs, you have to actually culture the PGCs to make oocytes in fetal granulosa cells. And then behold, 4 weeks later, you have mature oocytes. And with the male, you have to inject these PGCs into the fetal or the neonatal mouse testes, and then they colonize all of the seminiferous tubules, and you have normal spermatogenic stem cells. So just a simple outline of how you make an oocyte in vitro. I'm going slowly because even if I don't get all the way through this, I think it's a little intimidating to many people. But you should know that once you get this system, it's not going to be intimidating because we can purchase all these genes, and it's just going to be a simple formula for this kind of culture. So the primordial follicles are locked by FOX3 and other factors, and ovarian tissue cannot be cultured easily to an oocyte because the nature wanted these primordial follicles to be locked. So when Hayashi was approached by the German zoos that were trying to preserve the northern white rhinoceros, and there were only 3 left, and now they're all dead, he said, well, they wanted ovary tissue to be frozen. And he said, no, ovary tissue isn't what I really prefer. That's very difficult to work with. I just want skin biopsy, and we'll make oocytes from skin biopsies. And right now they already have beautiful PGCs from these extinct northern white rhinos. And I'm going to show you a little later in this talk how you can convert those PGCs with fetal granulosa cells into normal oocytes. So then that's 3 to 4 weeks of culture to turn skin cells into iPS cells, and then 2 days of culture to convert iPS cells to epiblast cells. And then epiblast cells to PGCs take 6 days, and I'll show you the transcripts. So PGC-like cells with fetal granulosa cells is another 3 weeks of culture, and that's called IVD, in vitro differentiation. Then FSH culture, amazingly the same in mice as you would expect it would be in humans. After dissociation of the granulosa cells, the IVG phase is another 11 days, and then just 1 day for exposure to HCG plus more FSH, and that's how you get in vitro maturation of these competent GVs into competent M2 oocytes that then are fertilized with wild-type sperm and result in healthy baby mice. So iPS cells can readily be made by incubating somatic cells like skin with just 4 known genes, which can be readily purchased commercially. One can use just 3 genes, but all 4 originally described genes by Yamanaka give the best results. And you can see KLF4, SOX2, OC4, MYC, and you can purchase these genes. And then once you've made the iPS cells, you can maintain them in just 3 genes, LIF, FGFI, and GSK2BI, with no feeder layer of fibroblasts necessary. Then you incubate these iPS cells in 3 different genes, again, all commercially available, to make epiblast-like cells. And those 3 genes are activin A, B, FGI, and KSR. You don't have to really remember all that. It's a cookbook, and you can just purchase those genes. And then the epiblast-like cells are incubated with 5 genes, and behold, you have in vitro PGCs or primordial germ cells. This is in the mouse, but it's a similar system in the human. And Amanda Clark has successfully translated this from Hayashi's work on the mouse to the human. Of course, there are different genes and it's species specific, but it's the same concept. So in the mouse, at least 5 commercially available genes are needed to do this, and they're readily available. And this is the original diagram from Hikabe and Hayashi's papers. And you'll note that it goes through the whole phase of going from embryonic stem cells to epiblast-like cells to PGC-like cells to oogonia to entermeiosis, all in vitro. And actually what you need is exposure of those differentiated oogonia to fetal granulosa cells. With adult granulosa cells, they'll just die. You must have fetal granulosa cells, and we'll talk about that in a minute. So in his early experiments, he made fetal granulosa cells. He was able to extract them just from fetal mice, but we're not going to be able to do that in the human. So now we've gone more sophisticated, and we know the transcripts that create fetal granulosa cells. And so we can use those transcripts in a separate culture. I'll talk about it later. Separate culture with iPS cells to make fetal granulosa cells. And so all you need to do then is take those two cultures, your experimentally produced fetal granulosa cells, your experimentally produced PGCs, culture them together, and you can see you get secondary follicles, expose them to 11 days, really 11 days is better than 14 days of FSH. And there are GVs that are ready for maturation in 11 days, and that just requires exposure for 24 hours to HCG with a little FSH. And this is just another diagram to run this home, how you go from in vivo, from an ICM to an epiblast, and then you have a PGG specification in the mid portion of that epiblast, and then you finally get PGCs. And it shows the genes below when you do it in vitro that recreates this whole embryonic process in vitro. And it's based on knowing and figuring by reverse transcriptate what are the transcripts that really are active, what are the genes that are truly functioning at this early embryonic stage to make germ cells. And this is just another diagram showing the same thing, comparing in vivo to in vitro. Now, in vitro, we'd have a serious problem of all these PGCs. When they're exposed to the fetal granulosa cells, they immediately go into meiosis and form secondary follicles and can be matured or die off. We can't hold them. They start working right away, and so it's a problem. Whereas in vivo, they're captured by the ovarian cortex, and that locks them so that they don't, they're arrested in meiosis, and that arrest continues throughout life, and it's just 1,000 a month out of 400,000 in the teenager that are gradually released over time until the woman runs out of eggs when she's 50. But it's still that primary fetal process of arrest in early meiosis. So the first 3 weeks of culture of PGCs and fetal granulosa cells would be about 4.5 months in the human, and that's IVD, and no FSH, and not sensitive to FSH. Then we open up those follicles and expose them for 11 days to FSH, and that's the in vitro gonadotropin phase. And I think we should be adding LH, just like in vivo, a small amount of LH to get better quality oocytes and a greater efficiency. And Hayashi is going to be working on this very soon. So use normal in vivo concentration of FSH and LH for in vitro oogenesis. It's just the normal concentration. The only reason for ovarian stimulation with high doses of FSH and a little LH, the only reason is to get the egg out of the ovary. And I talked about that last time, to form a follicle that's big enough that it ovulates, or you can retrieve it with a needle. But really, proper maturation occurs with just normal physiological levels. And that's what it looks like when we combine those PGCs with fetal granulosa cells. And then after 3 weeks, they have to be opened up. Otherwise, the exposure in culture, the FSH isn't going to get to them. Now, in vivo, the FSH gets to them just fine. But if you're going to do this in vitro, you have to open up those cells. See, I showed you those cells. You have to open them up in order for the FSH to get there. And then look at, lo and behold, you have beautiful metaphase 2 oocytes. And those are the first iPS cell-derived mouse pups from the skin fibroblasts of their mother. I was there with the C-section when they did that. It was really exciting. So in vivo oocytes require 4.5 months after recruitment and that's IVD. There's no sensitivity to gonadotropin, and IVG then for 11 days, just like in vitro, and they're ready for IVM. And this is a constantly changing daily dynamic. Oocytes that were recruited less than 4.5 months earlier are still going through IVD or early IVG. In vivo, once the oocytes have completed IVG, they are meiotically competent and ready for HCG or the LH surge. Constant daily process, and at any time, a certain number of oocytes in the ovary have completed IVD, IVG, and are ready for IVM. And this is the diagram I showed you last time that makes it very simple to understand what's happening really in the human. That you start with these primordial follicles, and after recruitment, which is occurring daily, and it's pressure mediated only, you have 4 months of differentiation in those granulosa cells. And then after that, you just have IVG for 11 days exposure to gonadotropin. And if it's exposed to gonadotropin for less than 11 days, then those ones aren't going to be mature. And if it's exposed to gonadotropin for more than 11 days, they're post-mature and they degenerate. So I'll just repeat again, you do not need ovarian stimulation for oocyte maturity. In vivo FSH creates enough GVs with meiotic competence, and you don't even need the normal ovulatory cycle for meiotic competence of GVs. You only need ovary stimulation or the ovulatory hormonal cycle for follicle enlargement and either monoovulation or for oocyte retrieval. So this is the group that works from 6 in the morning till midnight every day of the week to pull off this kind of work, which it would be impossible on a regular schedule. But these guys don't believe in a regular schedule. I shouldn't say guys. You can see that I'm an old man because I should say these people, because the women really were extremely key, and Ori Hakabi, particularly on the right of Dr. Hayashi. So what started all this originally was an unusual series of identical twins discordant for ovarian failure who underwent the first ovary transplants. And we started doing that in 2004. And we wondered why it could be that Crystal had fantastic fertility, eggs that were even good till she was 46, and Bonnie was born without any eggs. Why was that? So I talked to Amanda Clark, and who knows, I never dreamed we were going to get into in vitro gametogenesis with this. But she pointed out that if the reason for discordancy for ovarian failure is misallocation of germ cells at the time of splitting, we could determine in the human embryo when germ cell specification occurs by the obstetric history. And if we could generate germ cells from skin biopsy-derived iPS cells of these older women now, that would, both the fertile ones and the infertile ones, that would rule out an epigenetic or genetic error causing the POI, leaving the only explanation to be germ cell misallocation at the time of splitting. And there you can see, this is Bonnie, who, after a transplant from her sister, had 3 babies, and the last one she actually got pregnant with spontaneously, no IVF at age 46. So now both she and her identical twin sister are 55 years old, or were 55 years old. We did skin biopsies on both of them to see if we could make germ cells. And this is how we do the skin biopsy. And now it's routine on every menopausal woman or every azoospermic man. And so a 55-year-old woman who was previously fertile generated young PGCs from 55-year-old skin cells. But the twin who had POI and had no eggs generated just as many high-quality PGCs from her 55-year-old skin cells. And this is the paper. It took a long time to get this accepted. It went everywhere because I think it seemed too revolutionary and too shocking. But the study that Pandolfi did at Amanda Clark's lab was absolutely spectacular. I mean, it was so detailed. Every possible test was done to make sure these really were human PGCs, and that they developed in the same quantitative amount and the same quality from the skin of both the twin who had no eggs originally and the twin who had a normal number of eggs. But this was done when they were 55 years old and none of them had eggs. And this is just the diagram from where this was published in Cell Reports Medicine. They didn't quite figure it out right. When they made this diagram, they said results suggest a barrier to germ cell formation such that the infertile twin is unable to generate sufficient germ cells to overcome POI as a young adult, which reprogramming to human pluripotential stem cells can reverse. Not exactly. What it means is that there is no genetic reason or epigenetic reason why one twin had no eggs. It's clearly misallocation of germ cells. So this is from Pandolfi's paper, which finally got published in October of this year. But we had the data for this paper 2 years ago. Here we show that human pluripotential stem cell sublines generated from each participant make equivalent numbers of germ cells, equivalent numbers, there's no difference, and can generate their own amniotic sac-like structures using a stem cell-based embryo model. Using these stem cell lines together with genome sequencing, and they sequence the genome of all of these iPS cells, our data suggests that the lack of germ cells in the infertile twin is not due to a genetic barrier to germ cell specification or to amnion formation. Following reprogramming, the infertile twin cells are capable of generating human PGC-like cells and amniotic sac-like structures equivalent to her fertile twin sisters. So using these cells together with genome sequencing, our data suggests as a mild term, it really shows that POI in the infertile twin is not due to a genetic barrier and supports the hypothesis that during gestation, amniotic PGCs are likely disproportionately allocated to the fertile twin. And there you can see that all of our twins who are discordant for ovarian failure were monoamniotic, monochorionic. And that means that when they were just fetuses, their embryos split sometime between 9 and 12 days. If it was more than 12 days, they would have been Siamese twins. And if it was less than 9 days, then they wouldn't have been monochorionic, monoamniotic. So that means that we know that PGC specification has to occur in the human between 9 and 12 days. So we can skip these early steps with just 8 core genes now, which is pretty amazing. So you can take granulosa cells and with non-gonadotropin sensitive, they can make primary follicles that are FSH ready, and IVD. Okay, I'm just going through IVD and IVG and IVM, but all you need is 8 core genes to do the same thing, and I'll explain that. This is the Hamazaki's paper from last year, reconstitution of oocyte transcriptional network with transcription factors. So there are many genes. It's like in making a testicle. We know SRY does not construct the testes, but SRY is the trigger to many, many, many downstream genes that do construct the testes. And that's the same with these 8 core genes that are necessary for early oocyte growth. Again, you can purchase all these genes. And now it's important to know what they can do. These, we call them PPT8 genes, and they can recruit primordial follicles from ovarian tissue. Now get this is really important for fertility preservation. They can recruit primordial follicles from ovarian tissue to grow directly to secondary follicles, ready to respond to gonadotropin, that is to the IVG phase. It's just amazing. PPT8 genes are all you need to unlock the primordial follicles and let them develop to the IVG stage. Note, but this is what's different. The primordial follicles have already gotten through fetal granulosa cell stage. They have meiotic competence, STRA8 induced, and have undergone erasure of imprinting. So we can make primordial follicles go into oocytes with just these simple 8 genes, but that's not good enough to go from PGCs directly, because the PGCs have not been exposed to fetal granulosa cells yet. So all that the primordial follicles needs are these 8 core genes, and these oocytes are functionally competent to make normal offspring. But they can also be, these 8 genes can also be incubated with the stem cells or iPS cells with no preceding steps to epiblast cells or PGCs. You can just go straight to oocyte. It looks like oocytes. And these oocyte cells can be fertilized with wild-type sperm and cleave to a normal-looking 8-cell stage, but no further. And the problem is there's no meiosis, no imprinting, just oocyte growth. We still need fetal granulosa cells. We need more than the PPT8 genes. We still need fetal granulosa cells if we're going to start with iPS cells. So PPT8 genes and prior fetal granulosa cell exposure are all you need to make competent oocytes that can result in offspring. But we still need fetal granulosa cells if we want to go directly from the stem cells. So let's differentiate. There is the PPT8 is an abbreviation for these primary transition from primordial follicles, and diol is just an abbreviation from trying to go directly to oocyte-like cells from iPS cells, and you can't get functional oocyte-like cells that way. You still need fetal granulosa cells. So these are the three in vitro oogenesis scenarios summarized, and you can see that normal primordial follicles have already been exposed to granulosa cells, so you can make fertile M2 oocytes just from exposure to PPT8, those core genes. But if you're going to try to go from a diol, so to speak, and an iPS cell with those genes to secondary follicles and make M2s, without fetal granulosa cells, they will not be fertile M2s. So PPT8 genes are all you need to unlock the primordial follicles, but it's not enough. So regardless of this gene simplification, fetal granulosa cells are required to provide fully potent oocytes from skin cells. So the solutions for the human are to duplicate what fetal granulosa cells secrete to transform PGCs into oocytes, just to get those transcripts, or maybe somewhat easier, transform iPS cells into fetal granulosa cells by knowing what are the transcripts necessary in the fetus to make these fetal granulosa cells. So we still use the 8 core genes approach, reverse transcriptome of embryonic development from epiblast to somatic ovary, fetal ovary cells. And this was published by Yoshino last year, and not a lot of people are talking about it, but it's absolutely spectacular, because now in the mouse, you don't need fetal granulosa cells. All you need are skin cells, and you can convert those skin cells directly into fetal granulosa cells. And then all you have to do is culture the fetal granulosa cells that you got from one culture with the PGCs from a different culture. And lo and behold, you get normal oocytes that result in normal baby mice. And it's going to be the same process in the human. Amanda has perfected making human PGCs, and now she is very busy working on the process of making human fetal granulosa cells, transforming Hayashi and Yoshino's technology. And when this was published, Yoshino's paper, there was an exciting editorial in New England Journal of Medicine just this year, and this diagram from New England Journal of Medicine shows what Yoshino is doing, and what Yoshino is showing can eventually be done in the human. And you can see below there, you're just recreating nature. Differentiate fetal ovarian somatic-like cells into with PGC-like cells and culture them together. You get a reconstituted ovarioid, and they result in normal secondary follicles, which result in normal offspring. So I suggest you look at that editorial by Herbert et al., New England Journal of Medicine, because it summarizes in a way you can understand better how we're going to translate that to the human by making fetal granulosa cells just from stem cells. And this is sort of a summary from Yoshino's paper showing that the bottleneck to really making oocytes after you've made the germ cells is the supporting cells or the fetal granulosa cells. But you can induce ovarian fetal somatic cells from these PSCs and reconstitute ovarian follicles without embryonic cells. You can make them. And this is sort of a diagram that shows you how they were able with first transcriptase to find the specific genes at the different stages of mouse embryology, going from epiblast to nascent mesoderm to intermediate mesoderm to coelomic epithelium and the genital ridge to the fetal gonad. So based on reporter gene expression, a direct quote from Yoshino, based on reporter gene expression, we determined a series of culture conditions that recreate the differentiation process from pluripotent cells to gonadal somatic cells in a stepwise manner. Under these conditions, stem cells differentiated into fetal somatic-like cells, he calls them fossils, expressing NR5A1, a representative marker gene for gonadal somatic cells. through the nascent mesoderm, intermediate mesoderm, coelomic epithelial stages. And these fossils exhibit a transcriptional profile themselves and cellular composition similar to those in fetal or embryonic ovarian somatic cells or granulosa cells at day 12.5. And when these fossils were aggregated with PGCs derived from embryonic stem cells, the PGCs entered meiosis and subsequent oocyte growth accompanied the development of these fossil-derived follicles in culture. And PGC-like derived oocytes developing and the fossil-derived follicles were capable of fertilization and developed into live offspring. So these results demonstrate the constitution of reconstitution of functionally normal follicular structures fully capable of supporting normal oocyte production that can result in baby mice. And this is a picture of how he did that. And these are the baby mice. So our results demonstrate that functional fetal gonadic cells, fetal granulosa cells, can be induced from embryonic stem cells through a faithful differentiation process in culture, and this generated material may serve as a source to replace embryonic fetal granulosa cells for in vitro gametogenesis. And this is the summation diagram that Yoshino himself did, that functional oocytes can produce without the need of any embryonic tissue. And that's going to be very important for all mammalian species other than mice. We can apply this to the rhinoceros, which they're doing right now. We can apply this to the human. And I'm just guessing we'll have that figured out in about 5 years, but it's going to take a lot of maybe 5 or 10 more years of regulatory review before we're really going to routinely do this in the IVF lab. But clearly we have this platform now for, without going to any fetal cells from any human, we'll have to call it abortion clinic. All you really need are skin cells to make oocytes in 2 different culture systems, one to PGC and the other to fetal granulosa cells. And a similar thing is going to work for sperm, and Kyle hasn't done it yet, but that's Kyle's job. And so this is just a diagram showing the same thing in a simple way. From the mouse pluripotent cells, you can make fossils, and you can make PGC-like cells. You put them together, and you get normal oocytes that result in offspring. So again, this is another picture of the lab that's produced all this work. And I don't have enough pictures really of Amanda and her lab, but they are translating all this to humans. And our job really is to do the skin biopsies in the humans and to provide this material to Kyle and to Amanda, and also to obtain some funding to help with this and to do the translation in the clinic, because I'm not the scientist, but I'm just presenting to you what we now know from the scientists. So thank you very much for inviting me. I think this is one future, maybe in a decade from now, for dealing with women who lost their fertility, who didn't have the opportunity to freeze their ovary tissue or their eggs prior to their cancer treatment. So thank you very much for inviting me to this. I'm open for questions.
Very interesting talk. I just had a question about the health of the mice that you were saying. I think you said the Hayashi group has created thousands of live mice. I'm curious, like, do you know how deeply they've studied the health, both short-term and long-term, of the mice that are being produced this way?
I'm not sure I understood the question. We have a long-term study of the health of the mice, or I'm not sure.
Or just in general, like how much is actually known about the health of the mice at this point, coming from this type of method?
Well, so far, yeah, that's part of regulatory review when we do this in humans. But so far, the mice are healthy. They've been tested in every way, and they're healthy mice. I think, though, that I'm guessing from our human work that everybody knows in IVF that their original culture to make the PGCs involved no LH in the gonadotropin phase, not PGCs, but to make oocytes. They had these beautiful looking PGCs, but in the gonadotropin phase, they use no LH or HCG. And based on what we know from the human, I think just adding a very low concentration of LH will improve the efficiency more. But so far, I mean, it's a little bit of silence there, but I'll re-emphasize that the mice have been studied and they live a normal life and they're healthy and they're really having any problems with the mice. Amanda, I think, is in the human PGCs, is doing genome sequencing on every one of them, and everything's coming out normal.
Thank
Thanks.
you, Dr. I'm sorry, go on.
No, please go ahead.
I was gonna say, thank you, Dr. Silber, for this excellent talk. It is definitely very thought-provoking. What do you think in the United States is like the biggest barrier to having this become mainstream?
Oh, yeah. Well, because that's the question. What about, we're guessing, and you never know for sure, but we're guessing in 5 years, we'll be able to do this with those human cells. But when would we ever be allowed to actually transfer them and attempt a pregnancy? I think it's a good thing. Amanda is very, very good in this. We think to have this really strong regulatory review is really important, because the public is going to be scared stiff at first. And it deserves a huge regulatory review. Once we have the papers published and we show the technique and it can really be done, the regulatory review will be extreme. Once the regulatory review is done, I think you're going to find this is not difficult laboratory work, because you can purchase all these products. They'll be commercially available. Let me go on, because you wonder what is the stumbling block. As you know, we do have a stumbling block with either oocyte freezing or ovary tissue freezing. It's been very hard. It's better now than it was in the beginning. I started freezing ovary tissue in 1997. And it's funny, for social freezing, it's not all that popular, because women don't really start thinking seriously about this and until they're 38 or 40 and the eggs aren't very good anymore. And so I don't see that becoming a huge moneymaker. There are people that think it will be, but don't think so. I mean, maybe it will, but with cancer, it should be easy, because all of our cancer patients say they really feel fortunate they had cancer and survived, which is a startling statement. But they never would have frozen their ovary tissue or their eggs if they weren't about to undergo sterilizing treatment. So they had to make a quick decision, but they're really glad they made it. Now, so the only problem really is money. I mean, insurance needs to cover this, and I think it's really not a very expensive thing. If you go through a $250,000 or $500,000 cancer treatment, that's nothing compared to the just very minor cost for freezing ovary tissue, or I forgot what we charged, but we originally charged nothing because we wanted to make sure that money wouldn't get in the way of patients making their decision. And that goes back to 1997. But I think now the charges are still pretty low. The laboratory charges $1,000, and I think their total bill would be $2,000 or $3,000. And if insurance doesn't pay, then we forgive it. I think when insurance starts paying this, it can become much more popular.
Do we have any other questions? Oh, we have a question from the chat. Does patient's age play a role in making competent oocytes from skin cells?
I'm sorry, my hearing isn't so good, so repeat the question.
Oh, does the patient's age play a role in making competent oocytes from skin cells?
What a great question. I'm really glad you asked that. I have to emphasize this. No, the patient's age is not a problem. When you convert those 55-year-old skin cells to iPS cells, you're really just recreating embryology, and these are going to be very young eggs. Now, there will be mutations because there are somatic mutations that have occurred in those skin cells over the course of life. But they're not going to be that many, and you can easily just root out those mutations, and Amanda does that. And so it will only be PGCs or skin cells that don't have those somatic mutations that will be used. So they're going to turn out to be extremely young cells, and it doesn't matter how old the skin cell was. Results are the same in 55-year-olds. We have obviously some younger women with ovarian failure. And it just doesn't matter how old those skin cells are, the results are the same.
What are you seeing as the biggest scientific bottlenecks right now for getting this working for humans?
Well, the bottlenecks are manpower, because Amanda would have had these fossils figured out in humans. I think she'd almost be there by now. But it's really, during COVID, it became a serious problem because all the postdocs wanted to go into virology and funding problems. So we've got some private funding from philanthropists who are patients of ours to help the funding. But you need funding, plus you need to be able to hire the people that want to do stem cell work. And stem cell work was very popular 10 years ago. But now I think that one of the big stumbling blocks is just, I guess, enough money and enough support to entice young scientists to want to go into stem cells. I mean, they're being hired by private companies that are trying to develop vaccines, and that's all great stuff, but we really don't have the manpower. At least that's what Amanda tells me. She just doesn't have the manpower. She needs to move more quickly.
That makes sense.
Do you know if anyone is working on this in like a primate model or any other model besides mice?
Well, Amanda is going to be doing this in the primate model for sure. In fact, what I should say is the non-human primate comes before the human, obviously. She's made PGCs in the human, but now what she has to do is make PGCs in the monkey and make fetal granulosa cells in the monkey, and then make oocytes in the monkey and before she does it in the human. It'll be, I think, a pretty quick transition. Now, the person who really has the monkey work, and he's going to do it in the sperm, the person that really has monkey availability and knows how to deal with them, is Kyle. So we're expecting Kyle to do a lot of non-human primate work.
Wonderful. Thank you for that answer. Does anyone else have any questions today for Dr. Silber?
So it probably don't, or maybe you're a little intimidated by it. But I'll just say that understanding this in vitro gametogenesis is important, not just for making those competent oocytes from skin biopsy, but what it teaches us about how the ovary really works. And my talk last time about in vitro maturation just comes up, it produces for you a real good model of how the ovary really works, which many reproductive endocrinologists, as I see it in their practice, are not aware of it. For example, it's important before ovary stimulation in IVF to put the patient on some kind of suppression, like birth control pills or GnRH agonists or antagonists, because you really want to kill off the eggs, and it's happening every day, that have either been through, not enough gonadotropin stimulation or too much gonadotropin stimulation in vivo by their normal pituitary FSH secretion. So understanding that what I call the sweet spot, it came out of understanding in vitro gametogenesis. By recreating that embryonic process, we're able to understand how the ovary works much better. Furthermore, I didn't show this, but Hayashi was able to show that pressure is the major modulator of meiotic arrest and primordial follicle recruitment. And he was able to show that by putting these in vitro generated oocytes that normally would just completely go through meiosis and be gone, culturing them in a high pressure incubator. And in a high pressure incubator, they stopped developing, they were arrested. And when he took them out of the high pressure incubator, put them into regular atmospheric pressure incubator, they started developing again. So through this in vitro work, I think we have a much better understanding of how the ovary really works.
That's great. And are there any other questions? I just thought of a question. If you had a patient who had primary ovarian insufficiency from like Turner syndrome or Turner syndrome mosaic, and you were able to develop oocytes from their skin cells, would there be a way to select for create embryos that would not have the chromosomal abnormality?
Well, you know, that would be very tricky. Hayashi has done the experiments people asked about. Can you make oocytes from male skin cells, or can you make sperm from female skin cells? And the answer is no, it doesn't work. And so I guess with the Turner syndrome, what you're relying on is that the Turner syndrome girl would not even be alive if she weren't a mosaic. And a mosaicism could be so tiny that you just assume they're a normal exo Turners and they're not mosaics, but they have to be mosaics or they wouldn't survive. And so that's why there can be eggs in Turner syndromes if you get to them very early. I don't think that these eggs degenerate any faster than normal. There's a normal recruitment process that goes on, and if you start out with only a few eggs, because there's only a few cells in that ovary, or a few oocytes that are XX, they're going to be gone very quickly because there aren't very many. So, but I think going from a skin cell is going to be tricky, and it's going to rely on the fact that she has to have some mosaicism if she's alive. And so you'd have to pick out those cells that are XX. I think that'll be tricky, but that would be the way to go. But using the Johns Hopkins immunosuppression protocol, I must say that we're transplanting tissue. You know, there are certain groups of religions where they won't accept ovary egg donation, but we can transplant their sister or cousin's ovary tissue to them with safe immunosuppression now, and that really works. And they're menstruating and ovulating, and we're just waiting for a baby before we actually publish that.
Oh, that's great. Thank you for sharing that. Great. If there are no other questions, we can conclude this fascinating webinar that we had today, but I want to make sure everyone's had a chance to ask all their questions. Okay, it looks like everyone has. Well, thank you so much for Dr. Silber for this talk. It was exciting. I'd actually look forward to seeing it again, so hopefully I will in the future. Thank you everyone for participating, and you will be able to also watch this webinar. It'll be available on YouTube. Thank you.
Thank you very much, Mindy, for inviting me, and Mahmoud. Thank you.